Good morning, everyone. It is my pleasure to introduce our next speakers. I'll be introducing a group of scientists, and in no particular order. We have Arum Han, who is an Associate Dean for Research at the College of Engineering at Texas A&M. Next, we have Christina Acedo, who is a postdoctoral fellow working in the realm of tissue engineering. And finally, we have Ram Kumar Menon. He is a professor in the Department of Obstetrics and Gynecology at the University of Texas. Please join me in welcoming this group.
Thank you for the introduction. My name is Arum Han. I'm from Texas A&M University. First time being in Indiana University, so thank you for the invitation. As I walked to the campus, I was trying to figure out whether Indiana University and Purdue University is a rival or collaboration, but I do see a lot of a joint activity and joint building, joint grants. So I guess at least there is a decent amount of collaboration going on between the two universities. So today I'm going to talk about organ-on-a-chip of the fetal-maternal interface that potentially can be used to minimize the use of animals, which is, I think, one of the topics of this session. I'm an engineer by training. All my degrees are from electrical and computer engineering. However, throughout my starting from my PhD, I have been focusing on applying a lot of this engineering technology for biomedical application, most of them in cellular phenotyping, cell culture, and that has led into the organ-on-a-chip. So I have a mixture of a engineering-ish slide as well as some of the interesting biological results we got. I'll probably try to focus a little bit more on the engineering aspect because I think my colleague Ram Kumar Menon later on we'll talk a little bit about more of the overview of how this fits into the broader picture of where organ-on-a-chip can make a big impact. So a brief introduction to my lab. My lab is working on broad ranges of topic. A major focus is obviously an organ-on-a-chip device. We do also do a lot of a high-throughput single-cell assay, some of the synthetic biology application, et cetera. But in all of this application, the core of what we do is this microdevice, microfluidics. We also call it lab-on-a-chip or organ-on-a-chip. That's really the core competency of my group, and being able to apply this one to broad spectrum of a biomedical problem. We have several organ-on-a-chip projects. Quite frankly, before the term organ-on-a-chip came out, we used to call it a co-culture microsystem. So we have done quite a bit of neurocell co-culture devices, which then later on transitioned into working on some blood-brain barrier on a chip. And we have also done some GI tract mimicking chip model. And the past about 7-8 years, I have been collaborating pretty heavily with Ram, who is in the audience here, on our fetal-maternal interface development. Now, I realized that I actually missing a slide that introduces what organ-on-a-chip or microphysical system is. So just verbally briefly introducing this one, it initially, for the past 20 years, people have developed a lot of microdevices to enable cell culture, especially co-culture, and enable to position cells in a particular format that mimics the in vivo arrangement. And it could be anywhere from cell trapping, organoid trapping, et cetera. And so it's really a combination of engineered microscale devices with cell components in there that allows not only cell analysis, but also allows a variety of different co-culture. And obviously 3D bioprinting is also one of those micro technology that can be utilized here. And it really enables the cells to be arranged in a particular way that mimics the in vivo functions and the structure. So we call it organ-on-a-chip, tissue chip, microphysical systems. These are all interchangeable usually work. So I'm going to talk a little bit about the microdevice and how these are fabricated. Typically, these microdevices are initially fabricated, at least the master mold are initially fabricated in a clean room environment. These are clean rooms, basically a clean room that's commonly used in semiconductor industry. Most of the major universities have a clean room, and it has special tool, and this is where the initial mold or initial device is being fabricated. It does require specialty facility. However, the good thing for many of my bio and life science colleagues is there are a number of foundries out there that where if you send a design, they will actually make your initial prototype and device. And many of these university clean will also basically provide a foundry service. So this kind of technology now is much more available, whether you go into a research center or university clean room or you go to an external foundry. Once you have the master model, and I'm going to talk about this a little bit more, you can now replicate hundreds of thousands of these devices. So you don't need to have access to that complicated facility, but you can actually do your in-house rapid prototyping. What you see on the right side is this is a small part of our lab where we set up just essentially a cleaner room so to prevent any dust accumulation, and that's where you can do all of the device assembly. And then on these devices are compatible with regular sterilization technology, and that's how you can do your device testing. So we have, for the past maybe a decade or so, we have developed a variety of different organ-on-a-chip devices. In the upper left corner, you look into some of your neuroglia co-culture devices that basically allows two co-culture compartments connected by arrays of microfluidic devices. We have done some of the now blood-brain barrier on a chip. This is what you see on the left side, blood-brain barrier. You have basically a membrane integrated, and you have an electrode integrated to look at your membrane integrity and how it changes. You can measure electrically how the membrane integrity changes. The center part is what I'm going to talk a little bit more. These are the feto-maternal interface organ-on-a-chip that we have developed, and some of the 3D printed interfacial chip that also our NCAS colleague will talk a little bit about it. So mainly today I would like to focus a bit more on where we have been applying this organ-on-a-chip technology for biomedical application. In our case, it has been on the preterm birth and adverse pregnancy outcome. Obviously, for this audience, I had the slide here, but for this audience, this is a very obvious one, so I'll probably skip this one. Now, one of the things that I learned through the past, I guess, 7, 8 years working with Ram is preterm birth is a very complex etiology. Obviously, infection can cause it, but there are also many factors that are somewhat difficult to assess. So with him, we have been developing a variety of different MPS or microphysiological system models of a female reproductive tract. We initially started out by building an amnion membrane organ-on-a-chip. That was our very first MPS chip of the feto-maternal interface. And then we developed a series of devices, which I'm going to introduce a little bit more in detail. So kind of focusing on MPS model for pregnancy and women's health. If you look at the feto-maternal interface, it's a relatively complex organ system. It has a placental interface and it has a fetal membrane interface. And one of the challenge here is if you look at the illustration on the left side, it's a complex multicellular structure. So for people who are doing MPS model development, the first thing is, okay, how do we mimic this complex structure? How do we mimic the function of this complex structure? And then how do we mimic the responses so that when you're doing a testing on your MPS device, that testing result actually can be translated into a in vivo finding. So challenges of many of this current model is some of these are common to all of these MPS devices where 2D or explained human cell culture is insufficient. A lot of the animal models are insufficient. A lot of my cancer colleagues are saying, well, we developed a great drug for cancer in animal, but that doesn't translate very nicely to human. So how do we, that's some of the limitation of animal model. And of course, in the case of pregnancy, the difference is quite more stark. Non-human primates are one of the best model, obviously very expensive to do it, so you cannot really use it. And then explained perfusion study is also not adequate. And these are the variety of different reasons why we were interested in developing a MPS model of this fetal-maternal interface in general. Some of our first large grant that we got is we were part of this NCATS and NICHD co-funded clinical trial on a chip. So NCATS launched this clinical trial on a chip back in 2020. 10 teams received funding, And you are seeing 10 of this project. One of them is our project on a preterm birth maternal-fetal interface on a chip. And that's where a lot of our study, originally study, is stemming from. So if I'm looking at the fetal-maternal interface, we have been focusing on fetal membrane primarily, but we also have devices that focus on placenta. So if I'm looking at the fetal membrane procedure interface, that's how the interface looks like in the top. The placenta interface is what is shown in the bottom. And for each one of them, we have developed a simplified model of this one into what we call a microphysiological system. So basically has one maternal compartment and 3 different compartments for fetal cells. And I'm going to go into a little bit more detail on how the actual device function. And we also have created a Placenta-on-a-chip. And these two chips are the foundational technology for many of the studies that we have been doing. So I would like to talk a little bit about how our particular device looks like. So first of all, in many of these devices or many of these organ systems, you have multicellular structure. So how do you mimic this multicellular structure? In other words, how do you create a co-culture device in an in vitro setting? Typically, you can do a vertical co-culture, which is essentially what a conventional transfer membrane is. Transfer membrane-based culture is. You can also do a horizontal co-culture. We purposely decided to do a horizontal co-culture for various reasons. So in the case of horizontal co-culture, the basic principle is where you have, in this case, we have 2 cell culture compartments on the left and right. These are connected by area of microfluidic channel. So what this allows is horizontal co-culture connecting microfluidic channels are small enough to prevent cells from just passively going from one compartment to another. In other words, if you load cell type A into the left compartment, they don't just migrate, they don't just passively migrate to the right side. So you can confine these different cell type in each of the compartment. However, the area of microfluidic channel is still large enough for molecular diffusion to occur. So basically, you can think it as a transfer membrane, sort of a vertically horizontally positioned to separate out the left and right compartment, and that's basically enabled by this microfluidic channel. Now, you can actually fill this microfluidic channel with the extracellular matrix. So on the right side, you are seeing a scanning electron microscope image of a microfluidic channel filled by, in this case, collagen. And so these channels are few micrometer in height, a few hundred micrometer in length. So that's how it separates out these 2 compartments, but also allows the biochemicals to diffuse freely from one compartment to another compartment. And that diffusion can be controlled by the size and number of these microfluidic channels. So it's basically controllable depending on what level of a perfusion you want from one to another one. Now, these channels are actually large enough so that if we do see an active cell migration if the condition is right. Now, we need media because we want this co-culture to go on for several days to maybe a week. So we basically have a media reservoir sitting on top of it. So that media reservoir allows sufficient amount of a media to be applied, provided to the cell culture compartment. This is also where you can apply drug. This is also where we can apply environmental chemical toxin. And this is also where you can extract the media to analyze, for example, a cytokine production from the cell culture compartment. So what you see in the lower right side is initially we made many of these devices so that one device fits in a well of a 6-well plate. That has been our standard size. Okay? So basically it is a horizontal co-culture compartment. The main reason we didn't go to a vertical compartment is in the case of fetal membrane, we have more than 2 cell types. So if you only have 2 cell types, it is actually okay to do a transwell membrane type co-culture. But if you have multiple cell type, you cannot really do microscopy in the condition. So this is where the horizontal co-culture configuration is highly advantageous. So this is basically our basic model of all of our MPS devices I'm going to show here. Now, as many of you know, to really enable this microphysiological system or tissue chip, the cell source is one of the most important aspects. If you don't have the proper cell, you can make your plastic device all day long. It's not going to really work. So in the field of MPS, heavy focus has been on iPSC. So pretty much all of the NCATS-funded clinical trial, a majority of them are relying on iPSC. And in some area, iPSCs are more available. In some area, they are not available. Even when they're available, they are highly costly and takes a lot of time to properly differentiate. In the area of this fetal membrane, there's really no IPS available. So Ram's lab has been obtaining primary cells and had developed a variety of different protocols to make them into an immortalized cell. And his group has validated this cell type extensively so that we now have a very good cell source. And so initially when we first did the study, we used primary cell, but pretty much right now, immortalized cell are what we are using. Now, using this one, we have done a number of studies, and I'm not going to go into the biology too much, but I would like to just show a glimpse of the type of study that we have done and type of study that is possible. So the first one here is an example of a two-chamber MPS device, essentially a core culture type. And this is where we have, in this case, an inner compartment, outer compartment, connected by area of a microfluidic channel. And we have done a two-cell type core culture. And in this case, we have also looked into an active migration from one compartment to another compartment. And we use this particular two-cell core culture model for a variety of different studies. Now, one of our most extensively developed and studied model is probably a four-chamber fetal-maternal interface model. So basically has one maternal cell compartment and three fetal cell compartment, basically a ring structure. So we have center maternal cell and then three different ring-shaped culture compartment for fetal cell. And we have utilized this one for anywhere from mechanistic study to actually conducting a variety of different toxicants as well as drug study. For us, one of the interesting aspects of this one is several years ago, we started working on with our environmental toxicology group at Texas A&M. Texas A&M has a pretty extensive toxicology program, and they have been funded by the NIH's Superfund Research Center for a number of years. So we joined their center in 2022. So we are one of the biomedical projects in this Superfund Center where we're trying to use this fetal-maternal interface on-a-chip to look into impact of environmental toxicant on pregnancy. So this showing one study as an example. We have used this device to look at the toxicity of a cadmium. We picked cadmium as a very good positive control. The potential negative impact of cadmium very well documented. So this is one of our first controlled experiments we have done. So what this experiment shows is the big difference of you doing in just a 2D study versus a MPS study is in a 2D study, what you're seeing is you're basically looking into direct exposure to a chemical, and you can obviously assess the toxicity of that chemical to individual cell. But what you don't know in this 2D cell culture is you don't see the effect of how maybe an inflammation in one cell type then causes the potential negative impact on the other cell type. So what this one allows is you load cadmium, you apply cadmium only to the maternal chamber. So this is basically a maternal cell being exposed to cadmium. And what you can do is you can actually study how cadmium propagates from one chamber to another chamber. So that basically mimics, assess propagation of a cadmium or environmental chemical through different cellular layer. And you can then look at inflammation from each of these chambers. So we looked into how cadmium propagates from the maternal side to the fetal side. We also looked into how inflammation, in this case, we did a multiplex cytokine analysis, how cytokine responses in this chamber. And this allows us to look into not only a direct impact of cadmium, but also indirect impact. And here, when I say indirect impact, that will be basically a cadmium has not propagated to the other next chamber, but your inflammatory cytokines have propagated, and that in turn causes inflammation in the next chamber. So that kind of a mechanistic study is really what is enabled by this kind of a work. Now, despite all of this excitement where we are making hundreds of devices, we are testing hundreds of devices routinely. One of the it has many unique features, but some of the challenges are oftentimes they do require some dedicated tool and instrumentation. Not all of them. Workflow comparability from an engineering lab to a biology lab, sort of not there. User friendliness, sort of not there, and most importantly, oftentimes not high throughput. So if you talk with any of the toxicology colleague and you say, well, I can do a couple tens of testing, they say, well, come back to me until you can do maybe a couple hundred or thousands, because we are routinely using at a minimum 96 well plate, ideally 384 well plate. So that throughput is throughput and usability, and of course cost is some of the roadblock that is hampering the more widespread use of these MPS devices. So we focus on, can we really improve this one? So a couple of things. We focus on increasing the throughput so that we can sort of do a moderate volume manufacturing of these devices. So we did some variety of different design change. I said that you can, once you have what you call a master mode, you can replicate many devices. So what you're seeing here is from a small 4-inch single unit, we went into the array type so that you can actually have many of these, about 20 devices in a 96 plate format. So initially we were able to do only 6 devices per 96 well plate format. Now we have 20 devices. And applying this one to a 4-chamber device, we're going to still arrange it, do basically minor modification in design so that we can pack more devices in a standard 96 well format. So that's our first thing we have done here. So improve the manufacturability so that many more devices can be made and tested in the same time. Second is, can we adapt this device to an automatic testing workflow? So in our case, many MPS devices require flow, and if it requires a flow, you have to connect a complex tubing. In our case, in the case of fetal-maternal interface, we don't really need a shear stress. So a pipette-based operation is what we have been using. So far, we have been using just individual pipette, and anybody who has been working in the lab, if you have hundreds of devices, it means really thousands of pipetting steps, mistakes occur, time-consuming, etc. So how do we adapt this one to, at a minimum, to a multi-channel pipette, and ideally into a small liquid handling robotic system? So what we have done is we adopted this one into that 20 array model that I mentioned in the previous slide, and adapted this one into a robotic liquid handling flow. So we have used a OpenTron liquid handling robotic system. Those are usually on the 10K, so relatively affordable liquid handling robotic system. And what you see in the right side is that robotic handling system loading in this case just a color dye for demonstration. But what we have done is, we have now demonstrated that we can actually do cell loading with high consistency to all of these devices. We have done a media loading using this liquid handling robotic system, and we also have done a media extraction. So if I'm looking at this MPS device in general, initial fab takes some time. Then the time-consuming process is the cell loading part is time-consuming, and then every couple of days where you have to do a media extraction for later on cytokine analysis, that's the time-consuming part. So we are focused on how do you automate this one, and then once a prototype is done, then you can actually go into an injection molding system where these kind of devices can be outsourced and made in a large quantity. So basically, industry standard injection molding process is what you can utilize. And so in brief, a lot of these devices are becoming much more available to the broader user base than what it used to be just 5 years ago. So In the last maybe a few minutes, I'm going to talk a little bit about how we utilize this one, and I would like to show an example of a PFAS study that we have done. I think, again, for this audience, the potential impact of PFAS to human health in general, relatively well known, and there are also some epidemiology study on how PFAS might be affecting. Now, one of the challenges with PFAS is actually there are thousands of different PFAS chemicals. So when we say, well, PFAS is dangerous or PFAS is safe, kind of difficult to, because it's very much a chemical dependent. So what we have done is we have tested initially. We focus on, through our Superfund Center, we have about 56 PFAS chemicals that we are interested in. Initially, we pick 4 of them that you're seeing here, 4 to 6. We pick 4 of them, and initially make sure that they don't bind to this plastic to begin with. And so this is just some plastic binding study. But then we're showing that, hey, PFAS doesn't really bind to this one, which is a good thing. You don't want the chemical to be tested to actually binding to your device. That's not what you want. So minimal absorption. We initially did a 2D cell culture, 2D testing to do a dose range finding. So we basically applied PFAS, these 4 different PFAS to 2D cell culture, identify what is a sublethal dose but maybe cause inflammation. That's a good dose to apply. And so 2D dose range finding, and based on the 2D dose finding is what we then applied into our MPS device. So once we did a dose finding is when we are doing a direct exposure. So in the case of a fetal-maternal interface, we are putting the maternal cells in the center. So we're basically loading the chemical in the center compartment. In the case of a placental device, we're loading on the left side of the compartment, and basically doing incubating for 48 hours, and 48 hours is typically sufficient time for us to see a response. So we are then sampling from each of this chamber to look at to conduct endpoint assay. So we are looking at the viability, we are looking at a cytokine, and then depending on need, you can do a LCMS to look at how PFAS propagate across a different cellular layer. So what's interesting is we applied this one to both the placental interface as well as the fetal-maternal interface, because we really wanted to do a comparison and see how they respond differently to the same PFAS chemical. So in the case of a PFAS perfusion in a fetal-maternal interface, we can measure perfusion. We can also measure perfusion in the placenta interface. And I think if I'm just jumping straight into a final result, we do a viability assay using an LDH assay, because it's the easiest to do, where you just do a media sampling, and then you are doing a cytokine analysis. So in the case of cytokine analysis, we are using the multiplex cytokine assay kit. So it's kind of a bit busy plot to see, but we look into pro-inflammatory cytokine, and as we look at about 5 different cytokines of interest. And what you're seeing here is, in the case of a fetal membrane interface, limited decidual response, so that's a maternal cell. But in this, and there's a big difference in their response depending on what particular PFAS chemicals that we're applying to. In this case, PFDA induced the most significant fetal inflammation, especially in the AAC compared with other PFAS. So again, what you're seeing here is very much a PFAS chemical dependent response, where some we don't see much response, in some we do see quite a bit of response. And then we did the same study on the placenta interface organ-on-chip. Again, we are seeing some of the higher resistance as we're expecting from the placenta interface. And I guess the highlight here is using our fetal membrane organ-on-chip as well as a placenta organ-on-chip, we are able to look into impact stemming from a direct exposure. We can look at the indirect result from indirect exposure. We can look at the response of these interfaces against a variety of different PFAS chemicals, and we have a study plan to look into about 10 different PFAS, as well as their mixture. Our Superfund Center is focused on chemical mixture specifically. So look into individual chemical response versus mixture, as well as how these two important interfaces for pregnancy have a actually different response. And this one again shows the importance of having to look into this interface holistically to look at both placental as well as a fetal membrane. So I'm gonna sort of skip the summary actually. Some of the future direction for this one, I guess I already mentioned, individual chemical versus mixture is a high interest. We want to test a much more broader range of a chemical. We've been asked to, hey, we have about 100 different PFAS chemicals. Can you test it? And I said, well, 100 is a little bit challenging, but we can probably do a few tens of. But that's a big improvement from being able to test only a single-digit number of chemical. And of course, as you know, once you apply different dose, different time course, the number of experiments you have to do exponentially increases. And then ideally, population variability is of high interest. That somewhat limited to the number of cells we can get from different individual. And then really the last slide is we are going into some interesting, I guess, a translation of these devices. We are part of this Translational Research Center on Microfluidic system. And Ram will talk a little bit more about this one in the short talk, but it's basically developing, qualifying these MPS devices as a drug development tool by the FDA. And that's the purpose of this Trace MPS Center. And myself, Ram, and then Ivan Rusin, which is a toxicologist and who is running the Schupperfund Center. It's a basically three-team effort to qualify these MPS devices for by FDA as a drug development tool. So I think with that, I will stop here and obviously thanking all of my collaborators as well as the funding agency, especially from NIH. So with that, I appreciate your time. So I'm not sure whether we are doing a question right now or we're doing